Welcome to all. I am Dr. M. Armugam, Assistant Professor and Head, PG and Research Department of Botany, JJ College of Arts and Science, Autonomous, Pudukkottai, Tamil Nadu. Today, the topic of my presentation is research objectives. And when we define the research, it is the investigation of a scientific research problem in an organized manner where to gain the solution for the problems. And the main characteristics features of a research objectives are it should be very clear, concise, declarative statements, and also it gives new findings at the end of the research, and also it should be closely associated with research problems, and it has to summarize the achievement of this study, and it also has to be concrete statements describing what the research is driving to success or coming to say. And well worded objectives should be a very smart in manner, that is specific, measurable, achievable, realistic, and very time bounded. So the each and every words should be followed without any compromising in it. And the research objectives is a purposefully framed that can be reasonably achieved within the expected time bound and also the meantime availability of the resources. And as far as the need of research objectives is concerned, it will help to the scientific researchers in the focus of the study, and next one, it can avoid the collection of data, also organize the study in the stepwise manner, and it facilitates the development of research designs also, and also it orient the collection, analysis and interpretation, and also utilization of data where we collect in the research. And when we talk about the formulation of research objectives, it reflects that the questions those answered the investigators wants to study and also yield to a good result also, and it may be arised in the form of statement or questions. It offers the steering of ourselves during the conduction of the research. It always the scrutinizing as a guide in the interpretation of results. Then the research objectives can be divided into two categories. One is general objectives and another one is specific objectives. A general objectives describes the broad term what the study wish to accomplish, and the specific objectives indicates that how to accomplish the stated objects direct and the process during the research also. And the characteristic features of the general objectives are it should be a broad goals to be achieved or successed, and it is the study of a statement when the researcher expressed to achieve by his or her study. It is a commonly less in numbers. The specific objectives is concerned, it is a very short and narrow focusing manner. They are more in numbers but systematically arranged. Here the researcher should indicate that where, when, how and what researcher has to be carried out in our research problem. So, and here the few sample examples of objectives are given or discussed here. One is to formulate a reproducible and efficient protocol for the micropropagation of Curcuma pseudomondana from the explants of rhizome bud, rhizome and shoot apex as well as the leaf blades. And another one is to establish a standard protocol for the indirect organogenesis, that is callus mediated organogenesis from the explants of rhizome bud, rhizome disc shoot apex as well as the leaf blade explants of Curcuma pseudomontana. And third one is to standardize a suitable protocol for induction of somatic embryogenesis through careless as well as the suspension culture. And next, the extract of genomic DNA randomly amplification of polymorphic DNA analysis to identify the genetic variation of Curcuma pseudomontana growing in the different geographical isolations, and to understand the phytochemical analysis of the rhizome, leaves and also roots to establish the structural identification of the GCMS analysis, and to conduct antimicrobial studies to validate our traditional uses. And the last one is to evaluate the antioxidant properties of the extracts. So when we look into that, these general objectives of these examples we can discuss means, so from these objectives we can categorize the general objectives and specific objectives. Here the general objectives of this study is concerned, one is plant tissue culture studies and another one is phytochemistry study and also the genetic diversity of the species, and the last one is the screening of biological activity of this plant. These are the general objectives what we have discussed in the earlier sample objectives. And when we look upon to the specific objectives is concerned, that the specific objectives of this study is concerned, for an example, plant tissue culture study is the general objectives. Then it is clearly stating that direct organogenesis and indirect organogenesis, from the what explant we have to taken, what are the plants explants we have to taken, that is also discussed. Then somatic embryogenesis studies also be carried out, and in phytochemical studies the preliminary phytochemical screening also to be done, and GCMS study and able to the chromatographic also study we have to be conducted. And third one is likewise in genetic diversity study, what are the locations and where we have to taken, what markers has to be selected, these also has to be discussed in a specific objectives. And the screening of biological activity is concerned whether the antibacterial or antifungal or antioxidant or anti-cancer studies has to be carried out or not. And this, if you want to work out in this, you have to be indicate in a specific objectives. So likewise general specific objectives and framed on the research objectives. Thank you for the patient listening to my presentation. Thank you one and all.